Alright, so in this lecture, I'm going to be explaining on a survey on event extraction for natural language understanding. So this paper in 2021. The scientific literature embeds an enormous amount of relational knowledge encompassing interactions between biomedical entities like proteins, drugs, and symptoms. To cope with ever-increasing number of publications, researchers are experiencing a surge of interest in extracting valuable, structured, concise, and unambiguous information from plain text. With the development of deep learning, the granularity of information extraction is evolving from entities and pairwise relations to events. Events can model complex interactions involving multiple participants having a specific semantic role, also handling nested and overlapping definitions. After being studied for years, automatic event extraction is on the road of significantly impact biology in a wide range of applications, from knowledge-based enrichment to the formulation of new research hypotheses. So this paper provides a comprehensive up-to-date survey. So first, we establish a flexible event definition summarizing terminology efforts conducted in various areas. Second, we represent the event extraction, the related challenges, and the available annotated corpora. Third, we deeply explore the most representative method and present an analysis of the current state of the art. And fourth, we compare the solution applied in biomedicine with those evaluated in other domains, identifying research opportunities and providing insights for strategies not yet explored. And finally, we discuss applications and our envisions about future perspectives, moving the needle on explainability and knowledge injection. So, event extraction requires n-ary and nested relations, defining multi-relational structures. And it may concern many event types whose arguments may include an extensive range of entity type and semantic roles. Links among arguments. Event extraction may require the identification of links among the arguments of a same event. For example, a binding event type can take the following argument role type: theme, site, and site 2. But 2 is attached to different argument linkages. Events have different components, while natural language have semantic ambiguities, a variety of narrative styles. So the same accent or trigger might express different events depending on the sentential context. Similarly, an event might be described by completely different expressions and trigger words. Even within the same document, an event might be referenced by various extensions. Triggers can overlap by sharing a text span. So there are two kinds of events, three kinds. A simple event that it is not nested and no overlapping. But when there is overlapping, it means that, for example, one event is shared between two different, one entity can be shared by two different events, event 1 and event 2. Or in the context of nested events, for example, a trigger you have is connected to entity 1, but instead of entity 2 or mention 2, we call it there is another event which by itself has two entities. And so these are different types of, for example, I think 20% of BioNLP 1211 is nested events. long-range dependencies. And we have high-level linguistic phenomena. For example, here we have entity overlapping. In example A, you see the overlapping. Or in B, we see entity coreference resolution and double tagging. And these are problems that exist. So Genia is an anchor point for BioNLP ST series. Genia 11 events, for example, have properties not embraced by ACE25. For example, only 37% is nested, and 6% is intersentence, 36% sword separation between triggers and arguments. So you see that BioNLP ST13 and BioNLP, you have different data sets for them. For example, Genia MK, that uses it is same as Genia, and for example, cancer genetics, which is about cancer biology, which covers 600 abstracts, but the entities are 18 types, 40 event types. For example, if you go to GRO, it has 174 entity types. And you see different methods. Some people use graph neural network, BioLSTM, Cybert and graph neural network. For example, initializing Cybert for graph neural networks or any other approach. 3LSTM, but graph neural networks is more expressive than 3, is more general than that. But post-processing of methods like large language models is too long. And here we see the state-of-the-art accuracy for Genia, for example, is 63%, which is even worse than ACE25. That's why biomedical event extraction is more challenging than any other domain. And you see different data sets, different F1 scores, different methods. You either train it pipeline, like traditional pipeline methods, but if you do it jointly, then it may lead to better generalization